Hi everyone, I'm Ryan Calhoun. I'm here from the University of Western Ontario, and I'm going to be talking about a candidate gene for behavioral isolation between Drosophila simulans and Drosophila melanogaster. So in any kind of mating system, there's typically a lot of information that has to be exchanged and evaluated by both sexes. And the aspect I'm focused on, it's with respect to whether males are able to achieve copulation with a female in the first place. So typically males have a number of signal traits which they bear, and females evaluate males on the basis of these, and they usually have preferences that evaluate these males for one flavor versus the other for these particular traits. It's conceivable then that over evolutionary time, as populations of the same species start to diverge, that the mating regimes that they engage in start to diverge as well. And it's possible then that if a female of one population, if she were to encounter a male of another population, she wouldn't know how to interpret them signals, or would be actively adverse to those signals that that male produces. This is important in the context of speciation because the females that would reject mating with these kinds of males would cause a reduction in the gene flow between those two populations, which would cause new species to be formed, or at least the barriers between those species to be much more secure. So Drosophila is a very good system for studying mating behavior. It's been very well characterized, and males undergo this multimodal courtship system using a lot of different sensory modalities that females are assessing at each step of the process. To date, there are a number of genes that have been identified that influence mating at those different steps. However, the process I'm interested in, so the female recognition of other males, so female interspecific preference, there are not any genes that have been discovered yet. So a number of studies have looked at mapping different species pairs and where these different preference elements may exist in the Drosophila genus. The species pair I'm interested in is this one, melanogaster and simulans, where there's actually been mapping done to the level of individual loci. And the interesting thing about this system is that there's mating asymmetry between the reciprocal pairs here. So simulans females will not mate with melanogaster males at all. Melanogaster females, however, are much more permissive, and they'll mate with simulans males. That lets us ask the question, what happened at the genetic level in simulans that gave them this preference against interspecies mating that didn't happen in melanogaster? If you look at hybrids of these two species in the direction of the cross that we can get to go, so melanogaster females with simulans males, we see that these hybrids are like their melanogaster mothers. They're not choosy, so they will mate, which means that these simulans preference elements are acting recessively in these hybrids. So with that system, I've been trying to address these questions. So what genes are actively involved in behavioral isolation between these two species? What is the genetic variation that exists between these two genes? And what is that genetic variation spelling out in terms of the actual nervous system of or neuronal systems of these flies? So what neuronal substrates are they influencing? And ultimately, what are these females discriminating against with these different structures? So there's this great tool in melanogaster called deficiency mapping. So there's all these strains of flies where we have known deletions that have been generated. And that lets us uncover recessively acting loci in the opposing genetic background. So if we make hybrids of these, we can scan along this hemizygous simulans chromosome and ask whether or not we see the resumption of this simulans preference behavior. So if we keep scanning along, eventually we hope to find that there is a preference behavior, or one of these preference elements that we uncover. So just to give you an idea of the crosses, we have melanogaster females crossed with simulans males. Melanogaster females, we just represent the deficiency with that little black bar there. When we cross them, we end up with two products, hybrids that don't have any kind of deficiency, or hybrids that have that deficiency uncovering that, or making that region hemizygous. And we expect if we see the female preference that's present in D simulans, that we would see a reduction in mating. So we see that preference against mating with melanogaster males. We also have a control where we just do interspecific crosses, so melanogaster females with melanogaster males, and that's just to test whether or not, if we do uncover a reduction in mating, whether or not that's a reduction that's due to it, that deficiency causing a general ablation in mating behavior, and instead of an actual mating difference because of an interspecific difference in these males. So the first gene that we've characterized for this, that we've currently dubbed, is It's Not You, It's Me. And just to give you an idea of what kind of values that would look like, there's this. We've since followed up and found 3 different genes that look like they are involved in this interspecific preference, so not tonight here and not my type. Rather than show all the deficiency mapping and all these different graphs showing the reduction in mating that these females that have these different gene disruptions in them. Oh, I should mention, so deficiency mapping only works to a certain point. Eventually deficiencies are too big. You're going to capture 4, 5, 6, 7 genes, and so you have to move to another gene disruption system like p-element insertions or Minos insertions in order to actually test individual genes, but it works exactly the same way. So these are all the different p-element disruptions that we did on these different genes. Using g-tests, we got p-values all less than 0.5 or 0.05. Number of flies in each of these replicates was 30, 2 replicates for each of them. We paired each of these gene disruption hybrids with simulans males to show that these females are actually rejecting melanogaster males and not just every single male they encounter. And we were able to repeat this in 2 different strains of simulans for not you and not my type. So I'm just going to go into more detail about not you at this point. So previous literature has described this gene as a microtubule binding protein. It's involved in axonal patterning during larval development. It's highly expressed in the larval central nervous system, adult brain, ovaries, and there's a much greater degree of expression in females than males. Additionally, one of the key elements that we use has been used to disrupt this gene before, and when it's been used in disrupting that gene, it's shown that it has an effect in neuromuscular junction development, in dendrite elaboration, and axonal growth. To this end, this P element also fortunately happens to have a UAS element, which means that we can use the GAL4 and UAS system to try and reactivate the melanogaster copy of this gene in hybrids when we pair them up together. Moreover, we can use tissue-specific expression, GAL4s, so that this gene is knocked out in every cell, or the melanogaster version rather is knocked out in every cell except the specific tissues that this GAL4 happens to drive. And the hope is that when you do this, that you're able to see that normal melanogaster-like level of mating if you've targeted the correct tissue. So the tissue that we're trying to target is in the mushroom bodies. So mushroom bodies have direct sensory inputs, and they've been shown to be important in a number of decision-oriented tasks in Drosophila 4. So we picked 2 very non-overlapping GAL4 drivers to test this out in. And in the second one, we saw that rescue of melanogaster behavior. So it appears to be then this specific subset of the mushroom bodies, the gamma lobe and the core layer of the alpha and beta lobes that appear to be important in establishing this preference behavior. So that leaves trying to determine what actually are these females discriminating against. What do they detect in the males that they're reacting to? So there's a number of different possibilities here. And, I won't go into each of them. I'll just go into song because that's the one where we've seen the actual large effect. So the advantage of doing this in the context of our system is that if there are multiple elements that females are discriminating against, these are still all blocked by the melanogaster background, and because we have this disruption, that will let only that single mode of sensory perception, or sorry, rather sensation through. To that end, we can then modify or remove certain elements of melanogaster males in order to see whether or not the female reacts differently to them when they're only using this one mode of discrimination against these males. There's also the component of what are the females doing. So if these females are reacting to, so if these females have just a patent rejection of everything, but simulans males happen to have this magic key that lets them mate with females, so they are able to achieve arousal with these females, then we would expect that melanogaster males that lack the trait would show a reduction in mating. Similarly, if these females are readily accepting of mating, however, there's a particular mating signal that they do not like and they react against, then we would expect that when we remove that signal from melanogaster males will instantly start performing much better during these mating assays that we do. So the results in our two different strains of simulans that we found this effect for were that there was a very mild, negligible decrease in mating among the simulans in response to not having that signal. However, when we removed that signal from melanogaster males, we saw that the females showed a really drastic increase in mating with these males that they're normally adverse to mating to. And when we do this in the actual hybrids that have either the simulans-like behavior because we've disrupted that not U gene in them, versus the melanogaster-like hybrids, we see that we have that similar aversive mating reaction. So they suddenly aren't able to see the signal that they don't like anymore because we've stripped these males of that signal. And then in the melanogaster, we just see a general reduction in matings, showing that melanogaster just wants a wing song. It doesn't matter which specific wing song it is. So just to recap, so what genes are involved in behavioral isolation? So far we've been looking at, we found and are investigating these three, not U, NTD, and NMT. What is the genetic variation between these genes? Currently we've got different transgenic modifications in progress, and I can talk about those later if anyone's interested in what those are. On top of that, we've shown that there's differential not U expression in the mushroom bodies, and that establishes different mating preferences, and that these females seem to be aversive to the interspecific wing song that exists between these two species. So with that, I'll thank my lab for other funding agencies, and if you have any questions, I'll answer them then.